Hello everybody. Today we are talking about muscle physiology. The purpose of the lecture is about to describe the muscle function and major characteristics, including skeletal muscle, smooth muscle, and cardiac muscle at the tissue level. Also, we would like to describe the structure of a skeletal muscle fiber. Then, briefly, we would like to introduce neuromuscular junction and excitation-contraction coupling. We would like to list the factors that contributing to how much contractile force can be produced. And we would like to describe the types of muscle contraction and also briefly define muscle receptors. What is muscle? You can find different explanation. You can see that from Wikipedia, muscle is the contractile tissue of the body, and including the different functions to have locomotion and to survive, with heart muscle and then smooth muscle. And you can also find the definition from Oxford English Dictionary is a band of fibrous tissue in the body that has the ability to contract, producing movement or maintaining the position of the body, and it has power or strength. So these are the marathon runners, and here is the bodybuilder. So you can see that these people may have used different types of muscle fibers and they have been trained differently. So today's lecture, we would like to talk about how different muscles function and how do they relate to their structure. First of all, muscle function. As we all know that when you want to run, when you want to lift a weight, you all need muscle. So this muscle producing movement is the first function. And also when you're standing there, when you sit there, muscles maintaining your position, your posture. Muscles also stabilizing the joints. So with attaching to the joints, muscle stabilizing the joints. And when you are very, very cold, muscle also generate heat to keep your body temperature, that is shivering of the skeletal muscles. So with producing movement, this is done by all skeletal muscles, cardiac muscles, and smooth muscle. The movement by skeletal muscle is easy to be observed, but heart muscle makes the blood circulating around the body. Smooth muscle moves in the digestive system and also in the blood vessels. So these producing movement including all three types of muscle tissues. Let's look at the muscle tissue at the cellular level. So the first type we look at is skeletal muscle. The left side, you see a histology picture, which is being fixed and sectioned, observed under light microscope. As you can see that these purple, the dark blue one are the nucleus along the surface of the skeletal muscle cell, and there are multiple nucleus. This is a diagram. On the right side is a diagram showing that how motor neuron innervates the skeletal muscle with multiple nucleus on the surface, and both left side and right side picture has shown the dark band, which is called A band, and also the light band, which is called I band. So it's a very systematically repetitive structure that is very important with both skeletal muscle and cardiac muscle, and that is this feature we say that it's striation. And the striation, the base of them is the repetitive structure of sarcomeres. Skeletal muscle are the effector of somatic nerve system. What do we mean by that? We know that with a control system, you have to have a control center, you have to have a receptor, you also need to have effector, which is taking action according to the regulation center. So skeletal muscle is one type of effector, and this effector is being innervated by the somatic nerve system, that is, the voluntary movement. It produces voluntary contraction, so you know what you're doing and you do what you want to do. And this is specific structure between the connection of motor neuron and the muscle is called neuromuscular junctions. So that is very specific junction, how nerve command controls the muscle movement. And eventually it's multinuclear. That is because skeletal muscle fibers are being developed by fusing of multiple pre-muscle fibers. So if the muscle is being damaged, it's not easy just to have new muscle fibers in place. You have to have the cell differentiation, before cell differentiation, you have to go via the stem cells, then being developed and fused again. Now, let's look at the smooth muscle. On the left side, again, it is a histology picture under light microscope. On the right side, it's a diagram showing the illustrate the structure. You can see that in smooth muscle there is no striation, so the filaments, they are not very regularly arranged to form any sarcomeres. So it's quite random compared to heart muscle and skeletal muscle. These smooth muscle also are the effectors of nerve system, but it's effector of the autonomic nerve system, not the somatic nerve system. That is, you don't know when the muscle is contracting, and you can't control to let it contract or stop contraction. So it is done automatically by your autonomic nerve system. Therefore, the movement, the contractions smooth muscle is producing is involuntary. And the way that the autonomic nerve system controls the smooth muscle is different from skeletal muscle. So here on top here is the autonomic nerve system, and the connection between the autonomic nerve system and smooth muscle is by the varicocytes. So these are varicocytes along the neuron, and the way that the neuromuscular transmission is by diffusion. So it's not a specific like a neuromuscular junction with skeletal muscle. It produces neurotransmitter and then diffuses through to multiple cells in the skeletal, in the smooth muscle. And the way that smooth muscle cells are connected with each other is called a gap junction. So this gap junction is linking the different smooth muscles. The last one we talk about is the cardiac muscle. The cardiac muscle only exists in the heart, and it has some features with in common with skeletal muscle. It also has some features in common with smooth muscle. So let's look at it. The first one is appearance. the left side histology picture shows that you can see it's very regularly arranged with the striation, that is the sarcomere-based structure. And this is being illustrated on the right side with the dark band is A band and then the light band is I band, which is similar to skeletal muscle. And many other features are similar to smooth muscle. For example, it is cardiac muscle are the effectors of autonomic nervous system, and it produces involuntary contraction because you can't command when your heart is beating. In a way, you can modify it, but you cannot control it. The link between cardiac muscle also have gap junctions, and then these are the gap junctions, but the one that is specific to cardiac muscles are the intercalated discs. So the intercalated discs are the one that head to head between the heart muscles, and heart muscles have single nucleus. So having talked about the characteristics of these three types of tissue, we would like to concentrate on the skeletal muscle. So here, let's look at how skeletal muscle is being connected. If we look at the left side on top, we can see that the skeletal muscles are connected with the bones by tendon, and with the movement of the joints and the bones, so the muscle is possible to control, to contract. We would like to look at the structures in detail. So we take part of a muscle out and then look at the wrapping, the structure that is, so with a part of the muscle, we see that the connective tissue is wrapping around the muscle fascicles. So within a muscle fascicle, you can take out a single muscle cell, and with multiple nucleus on the top. This purple one indicates the nucleus. And with very regular arrangement within the muscle cell, so around the muscle cell is the sarcolemma, which is the plasma membrane of the skeletal muscle. Within the muscle, you have myofibril, which are the arrangement of the protein filaments. So let's look at the muscle cells in words. So here is the diversity of fascicle arrangement in the body. So the A type is a circular, so it occurs in the for example around the mouth, and then the B is convergent. So you have a point here, and then it's converging outside, so that is occurring around the shoulder here. C type is parallel. You have this type in the leg here, and the D type is unipennate, so it's one type of running diagonally. So this one occurs in the leg here, and then we also have the bipennate. It's occurring in the legs here, and the F type is fusiform, which is bulging, and this one is nicely occurred with biceps here. And the last type we have is multipennate. So the multipennate is around on the shoulder muscle here. So these are the different types of how the muscle fibers are arranged within a muscle. So within a skeletal muscle fiber, which is a single skeletal muscle cell, these are the structures that we would like to learn. First of all is the sarcolemma, which is plasma membrane, cell membrane. With muscle, you call it a sarcolemma because sarco means muscle. The second one is sarcoplasm, which is the cytoplasm, that is the fluid around within the muscle. And the next one is myofibrils, that is the main structure where proteins, filaments are being arranged. And you also have mitochondria, which provides energy. Sarcoplasmic reticulum is a smooth endoplasmic reticulum, that is specific structure within the skeletal muscle and cardiac muscle. Another one is transverse tubuli. This T-tubuli is very important in how the muscle can contract. So we would like to learn this in details. So in this picture, it shows that around the muscle fiber, that is the sarcolemma, and within the muscle fiber, the major structure where the thin filaments and thick filaments are arranged is the myofibril. And we have the T-tubuli, which is the transverse tubuli that is going into the muscle, that is important for the muscle contraction. And we also have mitochondria in between the myofibrils, and then the sarcoplasmic reticulum is storing calcium. So these are the functions, that the structures that are related to the function. And you can see here that is a neuromuscular junction where you have the axon terminal from a motor neuron, and you also have the motor end plate within the muscle. So we would like to talk about how the muscle structure is, and also what is the structure of neuromuscular junction. Within a myofibril, that is the one that gives the striated appearance, so that is striation with dark and light band repetitive structure. So one sarcomere is a repeating functional unit that is bordered by Z line. So with one Z line on the left and one Z line on the right, in between, that is one sarcomere. Within the myofibril, we have the thick filaments, which are the myosin, and the thin filaments are the actin. Both of them are type of specific protein. The A band is the dark band and I band is the bright band. So if we look at this diagram, we would see that on the left hand side here we have a Z line, and in the middle we have another Z line. So this is a zigzag Z line, and then in between them is one sarcomere. So with this one sarcomere, you have the dark band running. It's called A band. That is the length of the myosin. And then the light band is called I band. I band is the gap between the myosin. So here we have the thick filament, which is myosin, and then between the thick filaments are the gap, which is the I band. On the right side, on the top, we have a cross-section of A band, where the thick filaments are the circle in the middle, and the thin filaments are the dots around the circle, arranged in a hexagon structure very regularly. So that is how the cross-section look like. On this slide, the top middle picture is a diagram, and the top right picture and the bottom picture are both from electronic microscope with very high resolution. So let's look at the thin filaments in details. The thin filaments are also called the actin is the backbone. The actin is one type of contractile protein, and these are formed with G protein molecules and then with the F actin. So the double helical actin strands are called actin filaments, or the thin filaments. And then we have also the troponin as a regulatory protein, where troponin is here in the complex, the pink complex of protein. We also have tropomyosin. These are another type of regulatory protein. This is a thin thread, the pink one, and normally the pink protein, tropomyosin, covers all the black dots, which are the binding site for the myosin. So during rest, the tropomyosin covering all the binding sites for the myosin on the actin. So that is when tropomyosin and troponin are in place. This is like the top one is relaxed. So during resting state, the tropomyosin is like a door that closed with all the sides for binding the myosin are covered. So the door is closed. And troponin is like a lock. So when the lock is in place, the door is closed, then muscle is not contracting. When calcium ions are binding with troponin, then this is like a key. So when calcium ions open the lock, then the door of tropomyosin is opened. Then the myosin binding sites are exposed. So you can see that the pink long thread moved away from the black sites. The black sites are all available for myosin to bind. So calcium is the key, and troponin is a lock, and tropomyosin is a door. When the door is open, then the sites, myosin binding sites on the actin are now available. That is how the muscle is contracting, because when actin and the myosin are interacting, binding, then muscle contraction can happen. This slide shows the structure of thick myofilament, that is the collection of the myosin molecules. So on the top we see that there is a long tail with a helix structure, and then on the right side you have two heads. On top there are the two actin binding sites. So these two sites binding with the myosin binding site on the actin. The two purple sites are the ATPase site, where ATP has been hydrolyzed to provide energy. In the middle, it shows that the myosin has a long tail and then two sides. Each side has two heads. So that is how it's being collectively formed the thick filament at the bottom. So the sticking out ones are all the heads, and that are the cross bridges. In the middle, there is a bare zone. This is where H zone is. So let's look at the thick filament, how it's been arranged.
Next one is the cross-bridge cycle. So cross-bridge, when use one ATP and move a certain distance, that is muscle contraction, the basic cycle. So it can be divided into 5 steps. The first one is a binding of myosin to actin. So these two filaments start to interact with each other, and you have a power stroke. After the power stroke, it goes to the low energy state, which is the rigor. Afterwards, it's the unbinding of myosin and actin in the presence of ATP. Then it's the cocking back of the myosin head. This is a high energy state. So let's look at the cycle. The first step is the binding of a myosin to actin. So the head with actin binding site on the myosin binding to the myosin binding site on the actin, where you have both inorganic phosphate and ADP. When the inorganic phosphate is being released, it goes to the next step, which is the power stroke, where the head is changing the conformation and push the actin towards this side, the arrow pointing to the left side. So that is how sliding is happening. Then when ADP is released, it goes to the rigor. So this is a low energy state. If a person died afterwards, the muscle becomes stiff, and that is what rigor is about. And then in the presence of ATP, where if there's still energy, then the energy is ATP is binding to the head where the ATPase is. This causes the unbinding of myosin and actin, which is actin and myosin are detached. Then when ATP is hydrolyzed, so the enzyme is catalyzing the ATP hydrolyzation, then the head, this becomes the head of the myosin is cocking back. So actin and myosin are back to the next cycle, ready for the next cycle. So that is a cross-bridge cycle. Let's look at the details of the arrangement of filaments. On the top diagram, left side is a Z line, and right side has another Z line. In between, we have the myosin in the middle, thick filaments, and that's the golden one. In the blue ones are the actin filaments, and also the thin golden filaments are the ones connecting the thick filaments to the Z line. These are the titin. These are the connection protein. So at the bottom here you have electronic microscope pictures with the thick filaments and thin filaments, and in between them is a cross bridge that linking the thin filaments and thick filaments. So here are more electronic microscope picture showing that on top we have sarcomere, which is a thick dark Z line on each side. In the middle is the A band, that is the myosin with a dark zone, and on each side of the A band there is an I band, which is a gap that there is no myosin. In the middle we have H zone. During muscle contraction, it becomes the bottom picture. You can see that the A band stays the same and the I band is shortened. So the gap between thick filaments to the thick filaments in the next sarcomere has been shortened. So that is during muscle contraction, and that is how the sliding filament is about. So the sliding filament of the muscle contraction, acting on myosin filaments, they do not shorten, but the bands would have some changes. So during contraction, the A band remains the same because the A band is the same length as the myosin filaments. I band is the gap between one A band to the next A band, so I band shortens during muscle contraction. H band also shortens because H band is the gap between the thin filaments. So let's look at the animation. The filaments themselves do not shorten, but the band size might change. A band doesn't change, but H zone and I band shortens. So if we look at this picture, only number 2 is correct. A band stays the same and I band shortens.